Answering MCAT questions in under 60 seconds. Researchers are studying a compound called LX41, which modulates metabolic signaling pathways by altering hormone sensitivity. In muscle tissue, LX41 is observed to reduce AMP-activated protein kinase, AMPK, activation normally induced by insulin-like signaling. Which of the following best characterizes the action of LX41? So it sounds like they're saying LX41 is an inhibitor of this AMPK, which is normally activated by insulin. And what is AMPK, right? Well, AMPK is a really important modulator. It's a really important modulator of anabolic and catabolic pathways. Specifically, it normally activates catabolic pathways, okay? Whereas it decreases anabolic pathways, okay? And remember, it's a kinase, so it does so through basically phosphorylation of various other intermediates or enzymes, etc., right? And so it makes sense for it to normally be induced by insulin because insulin is actually a hormone that is going to promote certain catabolic pathways to occur, okay? Now, it also promotes anabolism, but in the case of insulin signaling for AMPK, it's specifically trying to upregulate certain catabolic pathways, like, for example, glycolysis. So let's look at these answer choices. A says LX41 enhances insulin action on the central nervous system. Well, again, we wouldn't expect it to enhance insulin action since it's blocking something normally induced by insulin, right? Nor would it happen in the central nervous system since this is specified to occur in muscle tissue, right? Which is not in the CNS, so that's wrong. B, LX41 inhibits the peripheral metabolic effects of insulin. And that sounds correct, right? Because it's blocking, again, something normally induced by insulin, so that should block the effects of insulin to some extent. C says LX41 increases insulin receptor sensitivity in skeletal muscle. Well, if it did that, it would be amplifying the effect of insulin, right? But here it's blocking something induced by insulin, so again, that wouldn't make sense. And then D, LX41 promotes glucose uptake by mimicking insulin. And again, it's not mimicking insulin because that would make it an agonist of something. Rather, here it's antagonizing basically an effector enzyme of insulin signaling. So that doesn't make sense, right? So B is the best answer here.